thank you very much for coming. I would like to know which background we have here. Like, what is your background? What you work on? Like, psychologist, or you are all psychologists? Okay, just to know how it is. So, I will try in these 2 days to I'll try to tell a lot of stories and show how powerful is transcranial magnetic stimulation or non-invasive stimulation techniques, which now you can use for all type of research. And it is like the next step after you are doing fMRI, EEG, or behavioral studies. This is the next step because it's really powerful and it allows you to see further what you cannot do with the other techniques. So I will try to share my experience with you. You can interrupt any moment you want if you have questions. The first day, well, this phrase is exactly how I see TMS, but still try for who knows what is possible. The problem with TMS is like some people get some results, some people get other results, but then the idea is always to continue trying if you can make it work, if you can get what you want. And the problem is like everybody does it differently, right? So I'm going to show you my way, how I have learned it, and then you will see in other labs they will do probably different. But so we will see how it goes. So in these 2 days, how the outline will be is like, today we will talk about the TMS principles. It's like a very important but boring part, I will say, like a lot of theory that you need to hear some things just to know, like the basics, how it started, like a little bit of history, neurophysiology a little bit, not to get you bored with this. And because to do TMS, you don't need to know it, but it's good to know it. Because when you are going to write your papers, when you are going to explain, it's good that you understand what you are doing. It's very important. Actually, this is really, really important information that you need to know. But okay, you will hear a little bit here, and then when you want to go further, you will read more. Yeah. So we will have first the TMS principles. That's super, super important. Is the safety aspects of TMS. And I think probably I will spend a little bit of time there because it's really, really important. Like, we are conscious what the technique can do and how it can affect other people, and how is our responsibility. And then in the afternoon we have hands-on, so that we learn how to do TMS, the basic stuff. I think if you do the course, you will be ready to start your study. I have envisioned this. So let's see. So tomorrow we will have TMS methods 1. It will be all, I think it's very important and is more fun, because we will talk about the protocols, all the protocols, well, the important protocols we have, how you can use them, just everything like based on research. I will have some examples of clinical studies. I'm not a clinician, I don't do clinical studies, but I have worked with patients. So you can use this to do research with patients and whatever. I will put some examples about therapy, just like 1 or 2, mainly used for depression, so that you see. And I think it's kind of cool. Tomorrow will be nicer. I am looking forward to it, because then I can talk a lot about my experience and all the experience you will have, which is kind of cool, like a little bit of important gossip, right? So it's kind of cool. And then it will be TMS methods 1, methods 2, so talking about all type of methods, and then TMS technique 2. And you can do any questions that you want. You can also, we can also do, if you want to, at some point, to have like some short talks to talk about a project that you have or ideas that you have and that you would like to make possible, so we can talk about it. Okay, so we start with the principles. So in this part, in this lecture, we will talk about a little bit about the history, the definition of TMS, and in what it's based on is the principle, and a little bit about neurophysiology. I will not get too much into it. I will just mention some stuff, so for you to hear the terminology. Then we will talk about really important stuff, that is the quantification of the TMS response, because this will be needed for every experiment that you do. And you should, the things that you need to take into consideration. I'm gonna, as I said, I will show you how I have learned it, how I think it should be done, but there are other ways to do it, yeah? So my way is not the only way, and not the right way only, so there are many ways. We will talk about the TMS equipment, because it's really, really important. The TMS equipment you have here are very modern, quite cool equipment, but it's different to all the equipments I have worked with. But regardless of the type of machine, you need to know type of machine and all the considerations that you need to know about the machine, coil type, and all this kind of stuff. And stimulation site. Okay, so we start. We all I don't know if it happens here, but in general, human beings have a lot of fascination for magnetism. I don't know if it is here is common. Like you see these people with the magnets in their hands, and they spend a lot of money with these magnetic things that they think the energy will flow better and they will be healthier. Do you have this thing here in Hong Kong? Like they use this? And well, no, it doesn't, but the fascination is there. And a lot of this had a boom in the mid of 19th century, when all the with galvan and all this kind of electric stuff came up. And here we have, in this part, here, one example of one of these treatments that is called general Faradayization. So this was used to cure hysteria and pain in the body and all kind of stuff. But somehow, probably this is the start of the TMS, like what then now we are using, right? Is this interest to know how we can use magnetism or electromagnetism to work, to help, to investigate, to work on the body, right? So here the patient is seated, and you see here are some pedals. So he has the foot on the pedals, and the machine is on the table, and the physician is passing the energy through connecting to the machine and the patient. So it's serving like a conductor to pass the electromagnetism for the patient to be cured. So it somehow started from there, from that part. And then here we see Thomson, which was a scientist, and he did one of the experiments that you see here. There are two coils. Oh, these two coils, and he applied some electromagnetism, and he could see flashes. So it kind of like it started there. Like he thought he could stimulate the brain, so he could see flashes, but probably it was not the brain, but that he just stimulated the cells, retinal cells, so he saw flashes. But you see all this fascination and all this work. He was not the only one. There were more people working on that, trying to create machines that they stimulated the body. And then the breakthrough, finally somebody got it and could stimulate the brain. And this was Merton and Morton. These two guys did an experiment with electrical stimulation. And they showed here, we can see something similar we will see here, motor potential. So he stimulated the motor cortex. So we see him here. And this is Dr. Adrian, and they are doing the demonstration to some students about how they could stimulate motor cortex. But this was done with electrical stimulation, and the problem was it was so painful that it could not be used for treatment or research. It was really, really painful. But anyways, they got it. They managed to stimulate the brain and then to move the finger that we will see. If you stay with me until the afternoon, we'll see. Do you have experience with TMS? Anyone has experience? Have you seen how it, well, yeah. Anyone has seen how it is? Quite cool. The first time I saw it, it was like I can move your finger and you don't have to do anything. So it's kind of cool. And then he got it there. And then he's showing there, and then this is part of the publication, when he was saying like, okay, it would be so cool if we could just electrically stimulate parts of the body and then it was like the breakthrough. But then it came the beginning of the modern TMS times that is now in the 85 with Dr. Barker. Then he designed a machine, or I don't know if he designed, but his team, where using electromagnetism or using a magnetic field, he could stimulate, do the other with so painfully. Now he will do it painlessly. It is painful, to be honest, but compared to before, it's not painful. Did you fill up the form that I sent, like the TMS form? Just to know if you are suitable, to know if you want to, who wants to try TMS actually? Anyone wants to try? I will not kill you, I will not make you more smart, although I would like to, because then we can try later, right? So for it would be nice to, like, you have the experience, how it feels, and anyways, so he started the modern times when he used magnetism, which will be cooler because you don't need the physics behind make things probably easier, and then it's less painful, and then he created the same thing, motor evoked potential in one of the motor cortex muscles, yeah, on the hand muscles. So what is TMS? So TMS is a non-invasive stimulation technique that is painless. I just want you to try, and then you say if you think it's painful or not. It is, yeah, well, it's like if I'm hitting your head like this, you know, for some people, and depending how you hold the coil, it could be more painful or less painful. Some people don't feel it. Depends on the threshold of pain that you have. But yeah, in general, it's painless, and it depends where you are stimulating. If you are stimulating in this area, it's less painful. But if you are stimulating in the frontal area, it's painful, because then you grab all these muscles here, and it's not even painful for the experimenters to see. When I see the people and they do like this, I feel, I don't know if to laugh or to cry, you know, you see it's so uncomfortable. But okay, if the person wants to stop, you can stop. So it's a non-invasive, painless technique that is used for assessing and modulating cortical excitability. But it's not, do you understand what it means assessing and modulating cortical excitability? So it means I evaluate the level of excitation of the brain, so I can know if I am more excitable or less excitable. Probably with the class we will be understanding what it means, right? Because all this technique is like the physiology behind that related to neurotransmitters and excitability of the cells and all this kind of stuff. So I am able to see if for healthy subjects, they have a level of excitability. For patient subjects, the excitability is different. So I can assess how different they are. And in motor cortex, it's very easy to see. It's kind of cool because you see it. You see it's happening. For other regions of the brain, it's a little bit more complex because you need to trust what other people are saying. Yeah, I do feel, or you need to see the behavioral responses, right? But in motor cortex, you see and you can measure how excitable the brain is, yeah? And modulating means that I can try to change the excitability, because one is a measurement, like when I put a thermometer, and I see, okay, you have 37 of temperature, right? This is assessing. I'm evaluating TMS. And the other is modulating. I can change it. I can make you more excitable. I can inhibit you. So this is the power of the technique that you don't have with EEG, you don't have with MRI, because with EEG and MRI, you are just measuring, you are assessing, right? And here, I am changing, I'm modifying. Some people claim they can make you more, like better for languages, or they can make you more like better, your memory can improve. So you can modulate. It is also good for neural conduction, measuring neural conduction, processing time, activation thresholds, which we will need for the stimulation. And the cortical excitability, we use the cortical excitability we measure could be inhibitory or excitatory. It's not only that I make you better, but I can also make you worse, which we will talk about it tomorrow. It's kind of cool. And is used to, before it was mainly, most of the work is done on motor cortex, because you can see, it's very easy to see. But now, and the last 10 years probably, there's a lot of studies in visual, somatosensory, cognitive functions because of the power of the modulation, yeah? And the combination with neuroimaging techniques. You have experience with EEG MRI here? Anyone, apart from you two? You have experience with MRI? But you know what MRI is? An EEG, electroencephalography. Okay, so now with the combination, as you have another way to see what is happening in the brain due to TMS, right? In other regions to study cognition, right? Motor cortex, you see it visually. I see it's happening. But to study memory, to study visual perception, you need something else to tell you, okay, yeah, there's something is happening, right? Okay, and then what is also very important is that it seems that TMS doesn't cause any type of long-term adverse effects. So I apply this stimulation to you, and in my experience, nothing happens. Because if it happens, so we wouldn't be doing research on it, right? Trying to make it better. So actually, people get scared about this, but it doesn't really change. The brain is plastic, so it's like apply the pulse and then suddenly you just change. No, it doesn't work like that. The brain just adapts to the stimulation you are applying. So it doesn't cause adverse neurological or cardiovascular or hormonal or motor or sensory, so you don't become more or faster, or you feel more from now on, or any kind of cognitive effects or health. Okay. What is the principle? Just a little bit of physics. I think it's kind of cool just to put the equation so it looks smart, yeah? Like people like just to see the equations. It's really simple, really simple. So the TMS principle is based on Faraday's law of electromagnetism, the electromagnetism induction. Very simple. Here we have a B is, you have a little bit of knowledge in physics? Well, you will have now. So we have here, B is magnetic field, and E is electric field. So this what we are saying here is a variable magnetic field. The magnetic field is changing with the time, yeah, and then it will create an electric field. Simple, really simple. Like, so we have here the figure. We have here the lines of magnetic field. The magnetic field is being created, yeah, yeah. So you have a current. For the magnetic field to be created, you need a current that is moving. You need a charge that is moving. So it's changing, it's moving. The magnetic field is being created, and then the electric field is being induced. This is all. Simple, right? So now how this applies to the brain. We have a coil, and this coil a current will come, and then a magnetic field will be generated. Here we have this is the current. The current is the red one here. And then the magnetic field is generated. And this magnetic field, as it's changing, will induce an electric field in the brain. With this, we have, depending on the coil type, that we will talk about it later, you can go inside 2 centimeters, 3 centimeters, depends, yeah? Don't take my word for it, but you can try and then see if you can get it. Sorry. So what is the sequence of events? What I said, you have an electrical current that is passing through the TMS coil. So you see it here. Then it's a short burst strong. It's not like a current that is passing. No, it's a burst. Boom, it's sand, right? Because it needs to be variable. If it doesn't change, it doesn't generate. So it is a strong magnetic pulse is generated and is produced and delivered on the scalp. And then as the magnetic field changes rapidly, it's very fast, it generates this electric field here. But the important stuff here is that the electric field will generate some currents that are called eddy currents. And these currents will be the ones that will affect my cells. Clear? Very easy, right? So here we have some in blue, some eddy currents. And these are my axons, the yellow ones. And then when the eddy currents interact with the axons, so there they can depolarize or hyperpolarize. They will change the membrane potential. Let me see if I'm forgetting something. No. This is basically what you need to know. So this current will change the neural activity. And that's the mystery behind TMS. Yeah. Okay. So where is this is just like an example to put everything like what is the big picture, what is happening with TMS. So we have a magnetic field that is applied through a coil, and an electric field is being generated, and then we have a macroscopic response that is behaviorally, my finger move or my finger move or my hand move, you can see that when you do TMS, or you can apply also in the leg, other parts. You have an evoked neural activity that you can see microscopically, or through EEG or MRI, as we have talked, or muscle twitches or changes in behavior. But it is the reflection of what is happening internally in the microscopic level, as we can see here. So the TMS is applied, and it affects the pyramidal neurons in the motor cortex or whatever neurons we are stimulating, and then it will do some contact with the axon, and depending on how the axon is located and how the electric field or the currents are interacting with the axon, we'll have polarization or depolarization or hyperpolarization. Here we have activation mechanism related to TMS, but seeing what happens on the axons, how it acts on the axons. And why this is so important, it's kind of, when I see, actually I put it there because I thought, yeah, that's important that they know this. When we're simulating an area, we need to know what we want to target. What the cells are located in a different position in the brain, right? So the effect of TMS for me will be different to you and to each of us. Completely different, because our brains are a little bit different. Yeah, obviously motor cortex is motor cortex, and dorsolateral frontal cortex, but how the brain is folded and is, our shape is different for each of us. So depending on how the electric field induced is approaching the axons, we'll have an effect or we don't have an effect. So here we have an example of an axon that is in an electric field that is uniform, so nothing is happening. And here we have the end of the axon, and then in this end there is a variation, and then there is an effect of the electric field, an effect of the TMS, yeah, because it changes only on the variations or the bending parts of the axon, where there are changes. Do you understand that? So if the thing is, if the axon is parallel, well, nothing will happen. It always will change when there is there are variability in the axon. Yeah, so we see here D means depolarization and H means hyperpolarization. And the best part where the induced currents affect the axon is in the bends or in the ends. When it's bending, so there is a change there, and then the electric field can do something to this axon. As I said, why it's important? You can apply the TMS that you want in any area, but if you're applying in the wrong direction, if the current is entering the wrong direction, if you are inducing in the wrong position, you will not reach the cells. It doesn't matter how much you affect. You understand this? So that's really important to know. So that's why the orientation of the coil, how I'm applying my TMS is very important. What the current, where is the position, so that my goal is to reach these cells that I want to stimulate, cells, X cells that I want. Clear? Let me see. So yeah, so keep in mind, that's why if the electric field is parallel to the axon, the activation will occur in the place where there are changes, where the intensity changes, okay? That's why in the end, there is a change, right? Because it's the end of the axon, so there should be a change there in intensity. So that's why in the end, or the bending part, there will be changes in the effect of the electric field. Okay, any questions to there? Okay, so how TMS affect the different regions is kind of complex. The area more studied, as I said, is motor cortex. And I'm going to just say how it acts on the motor cortex. And what is happening is you are applying the magnetic field, the electric field is induced, and then it creates like a series of descending volleys. You know what descending volleys are? I didn't know. I didn't know either. And it sounds so strange. Like a set of waves. You apply the pulse and a set of waves is being generated. Yeah. And this is really interesting, this part. And I always heard, when I just started, always heard these like the volleys and the waves, and like, yeah, I will study that later. It sounds a little bit like lazy, but it's actually quite interesting and important to know. So the interesting thing here is that these waves that are going to be generated, it will have certain timing. They will be generated each 1.5 milliseconds. The important thing is when you simulate with the electrical stimulation, the effect is completely different. So in principle, so the electric stimulation will generate some waves called direct waves, which I'll show here. So you see here, it means that the stimulation is affecting directly the pyramidal neuron. What TMS is affecting in an indirect way. So it is not clear why it happens, but it's believed that it is an indirect transsynaptic activation that is taking place there of the pyramidal neurons, but it's taking place there. So with the electrical stimulation, you do it directly. With TMS, you do it indirectly. So you apply, and 1.5 milliseconds later, the first wave appears. When you apply electrical stimulation, immediately you have a response. How do you know that? Because you have, they have been studied with epidural electrodes inserted that can measure these kind of things, and they have seen this type of differences. Now, with TMS, you can also generate direct waves, but you will need to increase the intensity of the stimulation. What this means, what it's telling us is that the electric stimulation had a different, the mechanisms are different for TMS and for electrical stimulation. So it's kind of cool.
So actually for electrical stimulation, it's actually direct. The current is direct, and actually we can modulate very, like if we have we can modulate the current.
Yeah.
How much we want to go through, but with the magnetic, it will produce a kind of electrical current. But this electrical current, how strong and how is there? Because due to this, I see there is a formula here. The one that we have given to our slide. Now we will show us a formula to calculate.
Oh, I'll reach there. Stocks in the second lecture.
Yeah, that one. So actually, there is a calculation there, right?
Yeah.
So how strong the magnetic field is, and we can also calculate basically how strong actually.
Well, actually, no. That calculation is not for that. But we'll get there.
Because just now we mentioned that the electrical current, if we use the TMS, is actually indirect. It's not
indirect, yeah, but you can. But if you increase the intensity, of the machine, if you increase the intensity of the current you are applying, you will be able to generate direct waves.
Yeah, this I know. So I also understand the basic idea that the stronger the magnet is, of course, the stronger the current will be. However, it's not like if it's electric, actually you can I can just by pressing the button or I know exactly how much current goes to the client or whatever participant. But this magnetic, so is there a formula that we can calculate?
Well, you can, there's no formula, no. But you have the, all these groups that are working with EFI modeling, right? You can model the electric field you are generating inside the brain. There are a lot of papers about it, and I think there is actually that you can calculate that, in the university, I know in the lab where I'm working now, there is a software that I think is free, is online, and you can access this, and I think, you can access and you just send some parameters. I haven't used it, so I'm telling you gossip, because I heard about this and they have told me, look, we have this, it's accessible for everybody. You just send some parameters, the software will calculate and will tell you what is the field that you are that you have inside. So, but I wouldn't be able to tell you like clear information about it, but it's not that simple as like, okay, I have just an equation and I put this thing here. No, I think it's more complex. And people that only work with this kind of stuff can give you more information about it.
Yeah, for me, I have a curiosity because I'm really layman. So, because actually we have the electrical stimulation and we also have the TMS. So, in comparison to the electrical one, so what is actually the major advantage of TMS if both of them is to create a kind of electrical current?
Well, for me, the advantage is that TMS, as it is a magnetic field, has the screening is less because of the skull or the properties. Yeah, but the electric field will have a lessening due to the characteristics of the tissue and the scalp and all this kind of stuff. So for me, in my personal, I work with both, but TMS is more powerful. Because, so and also different things, it's more focal, it's more focal, so depending on the code you use more focal, because when where you are using electric field, you are using an electrode supposedly here, and then you are stimulating all this area, and the current is passing there, and a lot of current is being lost, because the current will go through the path of the easiest path, right, that will be the skull. So a lot of current will be lost there. So what is actually reaching the brain? You don't know really, right? It's minimum. It's doing something because people are getting effects. But TMS doesn't have that problem. So depending on what you want, you might use the technique. Also, DCS is simpler. Electrical stimulation is a lot simpler, right? Like you can carry it, you can just put it, you can, but it depends. Every technique has the different advantages and the advantages and disadvantages. I prefer TMS. I think it's very powerful and yeah.
Thank you.
Okay, let's see. Okay, so we have here these figures here. We are inducing the descending volleys, and then it reaches the spinal motor neuron, and then it affects the peripheral nerve, then affects the synapses, and then suddenly I see a change in my twitch, and I see something like this in the screen, in my EMG screen, right? So I see like a sino wave. So here we see the excitatory circuit activated by TMS. The arrows are showing where they think in a model, where they think actually the TMS is acting, right? So initially, when you apply the pulse, supposedly, first it acts over this part here, and then an I wave is generated. And if I increase the intensity, 1.5 milliseconds later, I will have another later I wave. That will be, this is the I1 wave, then if I increase, it will be I2 wave, and then I3 wave, and et cetera, et cetera. If I increase a lot the intensity, I will be able to to generate a direct wave, which you can get immediately with electrical stimulation. But as I'm saying, it's because the mechanisms are different. They are acting over different parts of the cell or in a different way. Yeah. So here we see the epidural activity when it has been recorded with the epidural electrodes. So we see here how our I wave look, that the peak will be here, and then you have the later that will be 1, 2, 3, and then the direct wave which will be immediately after the stimulation. Here I'm just to give you an example of people that are working on this. No, there is constantly trying to get a right explanation what TMS is doing. So here we have two different models. This is one model and this is another model of trying to explain what TMS is doing. Yeah? I don't think we need to go into it. Okay. So, okay, yeah, I want to say something now that I see here. What is important, I'll come back to this part. What is really important here that is they have a very interesting property that probably you don't have with electrical stimulation, and it's that they have every wave has a different level of threshold activation. So as I said, you have an I wave, an I2 wave, an I3 wave. You need a different level of activation, right? And that's important because then you can manipulate better the stimulation, what you want to reach.
So is your stimulation process accumulative? So accumulative to
it is not known. So that's the part I want to come back to it. So it is not known if the I waves are generating because of the cells are getting this stimulation and then again and again and then they are firing. So it's not known because it's they are repeatedly firing, or because when you are increasing, you are stimulating different cell population. But the interesting thing is that when you are increasing the threshold of the intensity you are applying, you are activating or you are generating a different mechanism, right? You get a different response. So it's not clear if it is because they are firing and the same group. But something's happening there and somehow it gives some selectivity, at least in the response, in the outcome. We don't know how, but the outcome could be different depending on the threshold you are applying. And inhibitory interneurons have lowest threshold of activation threshold. Then the next one will be excitatory interneurons. So lowest threshold for inhibitory, then you need more threshold, more intensity for excitatory, and then pyramidal neurons need a lot more. Okay? So this gives you some, depending on your question, it gives you some level of playing with the stimulation, which is kind of interesting, right? It's kind of cool. So yeah, so the I waves is intensity dependent. And also, as I mentioned, it depends on other parameters of stimulation like the coil, or current, direction, and yeah. Other thing is not only with the level of stimulation you can generate D waves, it's really like a very deep, very wide field. If you change the correct direction, you can also, depending on the area you're stimulating and depending on the direction of the current, you can also generate D waves. Because obviously there, when you are changing the direction, you are acting over different cellular cell population. This is the important thing. So every response is a different part of the cell of different cell population. Yeah. Okay. So let's continue to remember A strong and brief magnetic field induces an electric field. The electric field causes neural polarization. Obviously, this is due to the currents that are being generated due to the electric field. Important, the magnetic component has no effect on the neural activation. Is that clear? We talk about magnetic stimulation, but the magnetic component doesn't have any effect on that. It's the electric component. So when you are using these magnetic things in the hand and all these things, you are wasting your money. When you're yeah, we are not magnetic. So the effect we are getting is not because of the magnetism, it's because the electric part. Okay. Nerve activation will occur in especially varying field or in homogeneous field occurs in nerve ends, what I show in the end when you have changes in the axon or changes like in the end of the axon or bending of the axon. Each time you have a variability in the axon, so you would have an effect you have an activation effect of the electric field. And TMS induces descending volleys. I waves are low intensity and the waves have higher intensity, but if you change, remember, if you change the direction of the current and you change the parameters, you can also generate D waves, okay? So that's why it's so important when you see you are going to plan a study, and it has happened to me that some people say, I cannot reproduce your study. And then I say, okay, so how did you do it? And they are doing it completely different to how I did it. So I said, well, you are not reproducing it. So if you want to reproduce a study and they use the current direction posterior to anterior, anterior, posterior, they did posterior, anterior. And you say, I'm going to reproduce it, but I'm going to use anterior, posterior. You will not get the same effect, probably, because the experiment is different. I'm not going to use the coil they use. I'm going to use a new fancy coil that I just found. You are not going to reproduce the effect. The shape is different. You are probably not going to reproduce the effect. So it's very important that you take all these things in consideration. And that's why I want to okay, I'm going to tell them this. It's boring. But we need to know, because from that you understand this will also depend your results, right? Your results depend on it. Like, okay, if I want to reproduce a study, try to copy really the study. It doesn't mean you will reproduce it, but you will be on the way. Quantifying the TMS response. I like this stuff more because it's more like practical stuff. And I have a lot of experience with it, of course, because I have done a lot of TMS on people. So how do you quantify the TMS response? We have motor evoked potential. I'm going to talk today mainly, and probably all the demonstration will be about motor cortex. But everything you learn about holding the coil and all this kind of stuff, motor cortex, you can apply it in another region of the brain. Why we do motor cortex? Because I can see what you are doing, right? So in frontal area, visual, well, I cannot see. I will have to take your word for it. I will explain you later about this. But motor, I can see. But you can apply. Obviously, as you were very well saying, you need to make some adjustments when you are changing the area of stimulation, and we don't know if the other region is going to react in the same way as motor cortex is doing. It means if motor cortex with the stimulation protocol is getting excited, so it's getting the motor potential is getting bigger, it doesn't mean that when I apply the same protocol in frontal region, I will get also excitation. Is that clear? So although we will talk mainly about motor cortex just for practical reasons, it doesn't mean that the effects are reproducible in other ear regions. The technique can be used in the same way. The effects, you will tell me when you finish your study. So we have different ways to quantify the TMS response. It's motor evoked potential, or MEPs, the different threshold we have. And we need to find first a hotspot, the spot where we want to stimulate. And I'm not going to talk about this. I forgot to delete this. Not today. I'm going to mention it tomorrow. Cortical silent period. I'm going to mention it because it could be interesting for some studies. But I think here people are not doing motor studies, right? It's mainly cognition for the psychologists. It's mainly cognition, right? Cognitive studies. And let's see if I manage to excite you about TMS, and then you want to use this machine a lot. Okay, so motor evoked potential and motor thresholds. So for the motor thresholds, for motor cortex, we use, well, that's why it's called motor threshold. We have different ways that we measure. When we were doing motor cortex studies, we always get a baseline measurement that is around when I get my MEP response. This is my motor evoked potential, yeah? How my muscle responds. This MEP will be, the amplitude will be here, and here will be 1 millivolt. And then I will try to get a region where I simulate my muscle, whatever muscle I'm using in the hand, and the response should be 1 millivolt. And that would be my baseline. When I do this when I want to test, for example, different methods. I want to know if one method excites or one method inhibits. So I want to know, I will have a baseline that would be 1 millivolt. I will apply a protocol, whatever I want to try, and then I will check if my motor evoked potential increases or decreases. It's simple, isn't it? Very easy. So you will be able to do that when we finish the workshop. But to do that, I need to also get the resting motor threshold. Resting motor threshold is when my hand is at rest, and the active motor threshold. This is the setup for like a simplified setup of TMS. So I have normally we work with figure of 8 coils. So there are many other coil shapes. We will talk about it. But here we have a figure of 8 coil, and it's very focal. So that's why it's used, mainly used. And I have a figure of 8 coil. I'm here putting my electrodes in the muscle of interest, the FDI muscle here. And then I have here my reference electrode. And then I will need to have a ground electrode, which I then do here. And then you have an amplifier that will amplify my signal, and it will go to my EMG. Okay? So motor evoked potential. What are MEPs? When you apply TMS over motor cortex, so the muscle responds. And the response is called motor evoked potential. They give information about cortical spinal excitability, because it goes, as we talked before, right? It's going down to the spine. And is widely used to study the physiology of corticospinal conduction in healthy subjects, and also in patients with motor pathologies. So it's really useful. So several parameters can be measured with the motor potential, as we said before. It will be amplitude, the amplitude. I don't know if I have an image about this moment. Yeah, actually I do. Here. Well, we have here the amplitude, that is peak to peak. Here's the TMS, the latency when it starts. So it gives information. It's not the same for someone who has a disease that someone who is healthy. The response is different. And also the area. So it shows the amount of motor neurons recruited in the spinal cord, amount of motor neuron discharging repeatedly, and synchronization of TMS-induced motor neuron discharges. So it's just this simple pulse, and the response, I get a lot of information. So,
So for people who, for instance, suffer from stroke, that part of their body that they cannot move.
Part of what?
Part of their body they cannot move. So can we actually use TMS?
Yeah, there are studies done for people with stroke, with Parkinson, and all this. Yeah, it's very useful. You should be able to see differences when you are, obviously if it's related to a motor area or whatever, when you are doing MEP, motor potential.
The stimulation is actually an involuntary behavior, right?
Yeah.
But does that mean this is involuntary treatment? So he has the behavior, and afterwards that will be good for his or her rehabilitation?
No. Depends on what you are. So there are two different things. One thing is when I'm assessing, I am trying to see what problem is happening in the motor conduction time and all the motor circuitry. And then I want to see, okay, it is fine, or it is damaged, or what is happening. One thing is that. Another thing is when I want to apply a protocol to make therapy. It's completely different stuff.
Okay,
So the therapy for people with stroke or with Parkinson, I don't know. You will have to check the literature. I have worked on methodology to see if in healthy you can improve. So you suppose, if I can do this in healthy, probably I will do it in patients, right? So I have worked in Göttingen, and in Göttingen we were working on like in different methodologies, trying to play with the simulation parameters so that we can modify motor behavior, right? To apply to people with different pathologies, including epilepsy or things that not only motor, but related to excitability. So there are different things. If you are applying a pulse in the single pulse over the motor area, doesn't mean the person will get more stimulated and then suddenly will be better. No. But this is a very, I think we will talk about it tomorrow, because then we will talk tomorrow a little bit about the concept of plasticity. Are you familiar with the plasticity? So motor TMS is a lot, it has to do a lot with plasticity. So we'll talk tomorrow about this, I think tomorrow. Okay, so all the information we can get from motor work potentials. And I'm putting all these things here because you could do a study with the thing we are going to learn in the hands-on, right? Because you get a lot of information. Of course, you are cognitive people, but I mean, you can be creative. I don't know. So I just want to show the possibilities that you have, because just with this MEP, you can see many things. You can create IO curves, like intensity curves, when you are changing intensity, and then you are seeing how the motor potential are changing and how they react. Yeah, so how the motor potential changes as a function of intensity. And yeah, in principle, this is the information I want to give you. Let me see. Yeah. Yeah, basically this, oh, sorry. So yeah, you have here different intensities, and then you see how the motor potential is increasing the amplitude. Okay, so motor thresholds. This is really, 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 really important, because all my studies, even for cognition, will be based on the threshold I find in the person. It's an individualized threshold. Yeah. So for me, I could use that is based on the stimulator output. Yeah. So what is the intensity, the level of this stimulator? For a stimulator will be different, but this stimulator with this coil, how much I need to apply for my finger or whatever to react, to activate the muscle, right? So for every person is different. For you will be 20, for me will be 60, for you will be 30. So for some people will be higher, for some people will be lower. Yeah. So that's really, really important, because my studies, the level of stimulation, the intensity I'm going to use, like the practical, I'm going to put a value. I'm going to apply this current to you, or this magnetic field to you, will depend on the value I have calculated here. Yeah?
Will the threshold change over the days?
interesting. It depends on the state of the person also. Many things have to do. If the person has been working more with the hands, or if the person is stressed, or if the person is anxious, or if the person has not slept, or anything, it will change the threshold. If the person is in a similar condition that we will talk about this also tomorrow, I will do in the end of the lecture a small summary, what you should take into consideration, trying to summarize everything we'll see here, because it's so important. People do sometimes experiment 8 a.m. the subject, and then next session 7 p.m. It should be different, probably, right? So it depends.
If it would be advisable to measure these water thresholds of every section?
Normally, depending on the study, normally the threshold don't change. But when I was doing my motor studies, we did it in every session. Every session we were measuring the motor threshold. It should not change so much. If you are doing the experiment, it will change, but it will not be like today you are 20 and tomorrow 60. That probably will not. It will have a variability, but if the person is in a normal, like in a very regular situation, right? You should not wait. You do the first measurement now in November, and then the next one in summer. It will not, probably will be very different, right? So you try. I would suggest this to you. We will talk about it tomorrow. I try that when I have several sessions with my subjects, it's just my preference, and then I will explain tomorrow why. I prefer that the person comes, if you come at 8, this session, you will come at 8, next session, and you come at next session. You have to be rested, you should be sleep very nice. If you are going to a party, call me, and you will not come, you know. Try to have the person in the best situation and that is similar to the previous situation. And normally, participants are very nice, and they try to follow normally these instructions. So yeah, I wouldn't expect, if the person is in the normal, like keeps more or less, things don't change, it should be similar.
I have another question. Here when we talk about the threshold, I still remember the fashioners described actually a kind of absolute threshold. That means actually the minimum stimulation that the participant can feel. And also he talks about the kind of threshold that is the minimum change that the participant actually can feel. Here actually the resting motor threshold and active threshold, I think is a kind of absolute threshold. It is the minimum.
I will explain what everything is. It's not what the patient feels, although I think I even know that technique. I learned it just recently. They like in a no way they were asking the participant, "Do you feel the finger move?" And yeah, I did. So just okay. So no. So calculating, asking the participant. I have heard and I read a paper about this technique, like asking the participant. We don't ask the participant here. We will not ask the participant. We have an EMG, we have a screen, and then we know what we want, and we check in the screen, okay, I need this this certain amplitude of my motor potential. I get it, yes, I don't get it, no. We will see it in the practical session, because it's very, this is like very subjective. If I ask the subject, if the subject replies to me, yes, it moved. I have had participants, I want to talk about it tomorrow, that they hear the coil and they move the arm without the coil being put on placed on the scalp. Just hearing the coil, because they have read what TMS is, and then you click it. They say like, so I said, no, you are not a good subject for me. So I don't trust asking the participants. I think it's better if you see. And you will see, that is easy to see. Yeah? So let's see what motor threshold is. Motor threshold is a parameter used to characterize the level of excitability in each individual, as we have said. So it gives information in research and clinical studies about the integrity of the corticospinal pathway. That it was something that you, I don't know your name. Your name was? Ruth. You mentioned, right? If the person has stroke or whatever, so my motor threshold will also give me information about the integrity of this pathway. And it's typically used as a measure that helps guide the intensity to apply during the different TMS protocols. So here in this definition, I'm summarizing all the things I have said before, right? So it's very important. Now, something again, I will mention this again, the threshold I find for motor cortex, this threshold not necessarily will be good enough for other regions, but we don't have any other way to do it unless using electric field modeling, right? You can try to calculate which one will be the intensity I need, but with modeling, software or things like that, right? But as everybody don't have access to this type of advanced procedures, so we use motor thresholds. It doesn't mean that because my brain reacts in motor area like this, so my dorsolateral will also react. Yeah? So keeping that in mind, we continue. What is resting motor threshold? So we have two thresholds for motor cortex. There are resting motor threshold and active motor threshold. So resting, without reading, what do you think is resting, as the word says? The muscle at rest, right? Just easy. And active? The muscle active. So very easy. So the resting motor threshold is typically defined as the lowest stimulus intensity able to evoke a motor evoked potential of at least 50 microvolts peak to peak. You remember this? I should have put actually a figure here to show you. So it means my MEP here, this amplitude should be from this point to this point here, 50 microvolts. Normally the finger wouldn't move there. So I don't trust, in the old times and some groups were doing it like this. They ask the person, did they move? It didn't move. So I say, okay, it didn't move. So you say, okay, yeah, so that's my motor threshold. But you can see it on the EMG, so you don't need to ask. I know that the amplitude should be 50 microvolts peak to peak, so I just check it. I will show you two ways to do it. I will show you two ways to calculate a motor threshold. One will be yourself, and the other way will be to use a software called PEST. And you can use both, any you want. Now I'm currently using the PEST method. But in the past, in Germany, I was using I. So I from the EMG, no? It's a technique called technique. You use 5 out of 10. You send 10 pulses, and 5 out of 10 should have the intensity 50 microvolts, okay? So here, you see it. So the resting motor threshold is typically defined as the lowest stimulus intensity able to evoke MEPs of an at least 50 microvolts peak to peak amplitude in 5 out of 10. So the person, we will see it tomorrow, or today. The person is there. Some people do it. We will learn it in the afternoon. The muscle should be at rest. At rest is at rest. No like this, no like this, no. Or like no, like this is at rest. I send 10 pulses, and I'm counting. Okay, if I am able to generate a 50 microvolts peak to peak, so I say, okay, yes, 1, 2, 3, 4, 5, 6, I lower the intensity. Back again, 1, 2, 3, 4, 5, until I get just 5 out of 10, okay? Or we use the other method. I thought this was kind of interesting to mention. The resting motor threshold and the age, which they are related. So I'm like, okay, that's like a gossip thing. I find it interesting, so I put it here. So the motor thresholds can be interpreted as a measure of normal electrophysiological maturation. So it gives some information about the stage of myelinization and the synaptic efficacy. So children will have a higher motor threshold, and adults will have lower threshold. So it shows the developmental, how the motor cortex is developing. Isn't that cool? But I saw it when I was working in Cologne the last years. The kids, we were doing TMS in children. And they need very high intensities for stimulation. And the motor threshold was really, really high, like really high. And I was kind of surprised, but it does shows the state of maturation, which is kind of cool. So at age of around 18, the threshold start is become like adults. So the threshold with age lowers the intensity. Yeah. Yeah, I thought that was cool to tell. So when you work with children, you will see that you need more stimulation for the motor threshold. If you work with adults, the motor threshold could be really, I have people that have just stimulator output 25, stimulator output 30, and with the kids, 80, 90, really high. So it's kind of interesting. Active motor threshold. So as we said, is when we are finding the threshold where the muscle is active. So we will activate the muscle. Yesterday we were doing some tests with Joseph. He was activating the muscle a lot. Did you feel fatigue? No, because some people get like, you need to activate the muscle. We were working with FDI. So unactivate the muscle and then people get fatigue sometimes. Muscle fatigue. So it's typically defined as the lowest stimulus intensity able to evoke motor potential of at least 250 or 200 microvolts. It depends. It depends on the definition. 250 or 200 microvolts. So everything again, 5 out of 10. I send 10 pulses, and from these 10 pulses, I want 5 that are at least 200. If I have 6 or 7, I go low on the threshold, on the intensity, and start again, 5 out of 10. So this is important as well to keep in mind. Motor potential and motor threshold are completely different. You understand that, right? So exam, to keep you like the question test, just to keep you awake. Why is the difference? What's the difference between motor threshold and motor potential? Nobody? You are no. So the motor potential is just the response of the muscle. I mean, motor threshold is like an intensity I need to evoke certain response at certain level. It's clear? So they are different. My MEP, I generate with my motor threshold, I can see an MEP. Yeah? The threshold are the intensity I'm going to use to stimulate, to guide my stimulation level. And the MEP is the response of the muscle. There are different things. The mechanisms are different as well. So some things to affect MEP, for my MEP to happen, are different to the intensity of resting motor threshold or active motor threshold.
So can you say that MT for a person maybe is, although it changes, but if a person has an MT, so it's relatively actually, there is a certain number of that, but MT actually any kind of stimulation will have a kind of MTs.
Yeah, you can have a very high motor potential. Motor potential is the response of the muscle. Yes, the muscle is responding. I apply 60%, 70% of stimulator output, and you see the response. But my motor threshold is the intensity I need for the muscle to be active at 50 microvolts. This is, it's a very particular definition. When my muscle is at rest and I just need to have a 50 microvolt peak-to-peak activation, what is this value? And they have different mechanisms. How do they know the mechanisms are different? So for generating MEP is different to get the value of my motor threshold is completely different to get a motor response. How do they know? Because they have done pharmacological studies, and with these pharmacological studies, they have realized that to evoke a motor, to get a motor potential, is completely different to when I want to get in my motor threshold. So the mechanisms are different. For example, the motor potential affected by GABA medication. So when they give GABA medicine to the participant, my motor potential gets affected, but my motor threshold doesn't change. So that makes you see that the mechanisms are different, and we are talking about different things. Yeah. Okay, I want to mention this, but I don't think I will make a demonstration about this because it's really difficult to generate. I was doing some testing before coming here, but it's really hard. So not only the motor cortex can give me a response, like behavioral response. The visual regions can also provide a behavioral response. The problem is that it depends on the response of the participant. Because with this stimulation, what I will try to do is to generate phosphines. You know what phosphines are? What is phosphines? Ken, you are Alex, you are the expert in the visual field.
So you see just the taxes of Chinese
And you hear it like flashes.
Good.
So it's the thing here is that it's very interesting because then you will try to find an intensity in which I am able to generate these flashes in the visual region, but I need to trust the response of this participant, right? Yeah, I saw. No, I didn't. So the person might think he saw, but he didn't, and then it's kind of difficult. I have done some experiments when I have seen the phosphines. Like you can really see them, but not everybody. It's very hard to get. And sometimes you need like really high intensities to be able to generate this phosphine threshold. But when you are working with visual system, it's the most appropriate threshold to use. But the problem is that you can really, you cannot get this in everybody. So if you read the studies in visual system that they have used phosphine threshold, they have measured 30, and in the end they have just 8 that could see this, and then you really see the population diminished because you cannot generate in everybody. There are many ways to do it. You can do it with single pulse, you can do it with double pulse. People have tried in many ways. So anyways, I think it's very important that you know it. It's very easy to do. It's just to put the coil in whatever region you want. You can try to get the phosphine threshold in the primary visual cortex, or the motion motion phosphine threshold that will be in the B5. That is the region for motion perception. You can try both. You will probably fail in both with most of the participants, but you should know that you have this possibility. Yeah. Currently, I'm working on a visual study, and we are using so far a motor threshold because it's so hard to get the phosphine threshold. So we are just gonna try with this. So phosphine threshold can be defined as the minimum TMS intensity that elicits perception of illusory flashes of light termed phosphines on a certain number of tracks. Same thing we saw before, but in the region of the visual region. The phosphine threshold is accepted as a valid parameter to determine the intensity using TMS visual perception studies. It's the more correct. Not everybody uses it. A lot of people use it, but not everybody uses it. Okay, we read Hotspot. This is a very interesting topic. I'm gonna tell, this is a gossip. So this is very interesting because the definition of hotspot is where I'm going to stimulate in the motor cortex. Yeah, I'm going to select a point where I'm going to stimulate over the motor cortex, and that will be my hotspot. And it's kind of interesting because the definitions, I have here like 10 definitions I'm going to show you, just because because, you know, when you cannot reproduce the study, it's probably because you are not stimulating the same region, or you are not selecting the stimulation point in the same way the other groups have selected it. So it's really important that you know this. And when you want to reproduce a study, and in the study they say, we simulated coordinate 20, 30, 45. And you want to reproduce the study, simulate 20, 30, 45. Don't go to 20, 45 minus 3, because it's different to the simulation point that you saw on that study. So why? It comes here with the hotspot thing. So I put here several definitions, because I did have, at some point, and I have had several times, some discussions about this, how we select this point. What is the point I'm going to stimulate? And some people define this as just the highest response. But the problem is that TMS is so it can have so many variabilities. If I talk, the MEP can increase. If the person moves the muscle, which I saw, I have worked with primates. And when I was doing TMS in primates, I always noticed that sometimes my monkey was doing like generating very big MEPs. And then I was holding the coil on the head, and like, what is that? And then I noticed he was doing like this. And I could feel it. And then each time he was doing like this, I was getting a very big peak. So I realized that probably was also happening in my human subjects. So I asked them not to do that, because sometimes you could see really big peaks, and then what is happening? So the definition for me, the definition of the highest peak is not the best definition. Because if the person is falling asleep, you will get very big peaks as well. And I know when the person is falling asleep, you will get a very big peak, and then a small peak, and then big peak, big peak, and then a small peak. So I wouldn't define it like this. So here's what is motor potential according to Rodwell, which is one of the big persons in TMS. The site of maximal amplitude can be called the optimal position, he says. The site of maximal amplitude can be called the optimal position. But it's not like this. If I move a little bit the coil, so I will get also a peak. It's like probably because you're stimulating new cells or whatever, and then you get this peak. So I have here, the coil must first be placed over the most effective point on the scalp for eliciting any response at all. So initially what you want to do when you are finding the hotspot is to find a place like you can see a response. This is the first part. You will not find immediately the hotspot, but you will find a place where you will see the finger move, and you want to find that place. So you start to search around which one is the stable part. So okay, I like a lot this definition that maybe you cannot see very well, but say once the MEPs are seen to be reproducible, consistent, and showing the greatest amount of activity, this position on the scalp was recorded at the hotspot. So very important in here that I'm able to reproduce the response. Not the highest response, sorry, but also that is reproducible. It's clear? So my hotspot will be actually, I didn't put a high definition of a nice definition of hotspot. Okay, I will tell you with my words what the hotspot will be. So the hotspot will be the place I can find a response, and the response is stable. You are able to find it. It takes time, but you are able to find it. If you are going to do motor cortex study, it's very important that you really have a stable point, because if I have a lot of variabilities, it's not my hotspot. If I want to test a method, if I want to have a new protocol, right? I'm just going to try a new protocol. I'm going to create some excitation that will be the cure for X disease. So I want to try this new protocol. I need to test it over a region that is very stable, that each time I send a pulse, I get a fairly similar motor potential. Is that clear? So if I get in that area, I select a region, and then I get a very high peak, and then the next 3 pulses I send, I don't get any response, this is not the hotspot. My hotspot is a spot where I send 20 pulses, 10 pulses, 15 pulses, and I'm able to get a response that is fairly the same intensity, the same amplitude. As we get a baseline, that I talked before about the baseline, there was 1 millivolt, so it means my amplitude of the motor potential will be roughly 1 millivolt peak to peak, 200 plus minus. So it could be 800, 1 to 750, but that you see that the response is constant. If you don't find that spot, so I don't think you are on the hotspot. Clear? But for cognitive studies, when we're working on motor cortex studies, it's very important to get like the real hotspot, because all your study will depend on that region, right? If I have a very unstable region, so I will get whatever response of my protocol that I'm gonna test. But for cognitive studies, you try to find also this, but you don't need, as we don't know actually, this is being recorded now, so. But you try to find your stable area, but obviously you don't want to spend a lot of time trying to find, because it could take 20 minutes, it could be 30 minutes, right? So, but you try to find a stable area where you are getting a stable response. You can get that with few pulses, and then from there you start to find, once I have my hotspot, why it's so important, I'm going to use this point to find my motor thresholds. So if I have a very unstable region that sometimes answers, sometimes don't, I will need a lot of intensity to be able to produce a motor potential. Do you understand this? So when I'm on my hotspot, I will be able to have a fairly stable response, and then I can find with more confidence my threshold that probably be lower than if I'm simulating a region that is not the hotspot. So we'll be trying to be close to the hotspot.
Another question. So actually for individuals, the motor threshold could be different. However, for the hotspot, actually there should be similar. Can we say that for the hotspot? So if, because the hotspot, because we say that the motor threshold is individually based, so everybody's could be different. However, the hotspot for a certain kind of motor reaction, motor response, will it be actually more or less similar, or is also different?
Yeah, because it depends on the head, size of the head, on the head of the person. So the hotspot is the location, the point I'm going to stimulate. So for Joseph, it will be more lateral. For someone who has the head a little bit bigger, so will be probably more central. Or someone who has, it has happened to me, like you have people that have like the forehead very big, so you think it's somewhere here, and actually it's more frontal. So the hotspot is where the point I'm going to stimulate. In the brain should be, it's equivalent to the motor cortex. The motor cortex is there, but the position where the hotspot is where I'm going to put the coil. And to get that, I will give you some guidelines. How can you start to search for it? But it doesn't mean it will work for everybody in the same way. So you will have to locate the coil a little bit different. You need to go a little bit frontal. It's completely different. But in my experience, once you have worked with one person, or you have played with try to put it next time, it will be very easy to find. But and then you get experience. You see the head, like, okay, it would be more frontal, it would be more lateral. When you don't find it, because sometimes you are looking for it where you will normally look, you start to move to other regions, and then you find it. So for every person is different.
Because last time when somebody showed us how to use this machine, and we see a kind of a physical movement, and it seems that the technician who was demonstrating that didn't take quite a long time to find it. I think that and then we can see the movement already. So I was thinking that maybe the hotspot could be quite.
What did he just had the hand like this and he found the spot?
Yeah.
And he was with someone he trained before, or was someone new?
Yeah, there was a technician. He actually showed how
in one of you.
I just asked one of the participants.
Yeah, so.
Show us, and we see the balloons. And
oh my God, it's very high, the bar then now. I have to find the spot like this.
I didn't see that he used a long time to achieve that.
He was lucky. Yeah, I will show you some way that you can find. Normally, it doesn't take time, a lot of time when you are experienced. He probably has a lot of experience doing it because he's doing demonstration constantly. Yeah, but it's not always like this. You might have people that you just don't find it. It is so. But I will show you some way that you can try to find it quickly, because motor cortex will be more or less in this area here. So one third from seat set, you know, using the 1020. So we'll be somewhere here, probably between this part and my seat set, somewhere here. So I will do something like that, and I'll put my code, and normally it will be somewhere there, not the hotspot. That doesn't mean the hotspot. I will be able to move the fingers. Now I don't know if that's my hotspot.
So that's the area?
Yeah, that's the area.
But then we can find the,
yeah. So now it depends on what you want. If you want to have just an intensity, the brain response, so you don't need to really be focused. Okay, it has to be the hotspot. But I know with this intensity, my brain responds, right? So with 40, I'm able to make it move. Now I will find in this region my resting motor threshold. It depends how precise you want to be. I think it's very good to try to find, in my opinion, it's very good to try to find a stable place, like you can reproducible, and then you get there the values. But he will not always be that lucky that he gets a person, he pulls the coil, and the finger moves. That one, second, sometimes it has happened to me. I see that the person doesn't move the finger, but you can see the MEP on the screen. So when you don't see the movement there, you should see the screen, and then you see, okay, yes, I do get some response, but for some reason the finger doesn't move. And I have seen some people, like they are doing demonstration that they just can't get it, and what they ask the person is to activate the muscle. And then activating the muscle is easier to have an MEP. But you will have some guidelines to start, but not everybody will be responsive to that guidelines. Some suggestion for correct hotspot localization. Well, correct, what I call correct, but as I say in other labs would be different. So just what I think is a good way to start. It's quiet environment during MEP recording, because when I'm doing I'm trying to find my hotspot, if someone passes in front or talks or something changes, so I get a peek. So you don't want that. You don't want the person so people around be talking or opening the door or closing or no, you don't want that. You want a quiet environment that the person is relaxed. Avoid muscle pre-innervation, because when the muscle is active, as I explained before, you need lower intensities. So it's very easy to generate, to have a response. You don't want that. You want to use the lowest intensity possible to do stimulation. Yeah? So for safety reasons, and yeah, so we want to have the best conditions for the participant and for our experiment. So take into account stability besides MEP amplitude, stay longer than one pulse on a selected spot, because I have seen that some people just get put the coil and click, they get a response and then change to other place and then change to the other place. No, stay there. If you find a response, stay there, check 2, 3, 4, 5 times. If you can get the same response, then search around, and then you will say, I don't know, actually this is the best position, and then you stay there and this is the spot. Yeah? If you get a response, stay there and no 20 minutes. Stay there, assess that this is the spot, and then like, okay, yes, I get a stable response. Let's check if here I get a better place. To get low, the idea is always to have the best region with lowest intensity from the machine, right? We don't want to apply 100%. We want to simulate with the lowest intensity possible for safety reasons. Once a stable spot has been found, recheck that is actually the spot by measuring again. So you say, okay, this is my spot, just put it again and check 2, 3 times. Oh, yeah, that's true. For cognitive studies, for motor cortex, you shouldn't do that. For motor cortex, you should really do like a proper baseline twice, that you can reproduce it, that you have 1 millivolt, that the standard deviation between the pulses is not big, yeah? And ideally, this should not take longer than 20 minutes. When you are doing this, you don't want to stay long time with the participant because the TMS is tiring. So with the time, the person starts to be sleepy and be tired and this. So you don't want, if you don't find it after 20 minutes, just let it go and try again next day. It could be for many reasons. The person is not the right day or is too anxious or whatever. With anxiety, probably it will be faster to get the motor potential, but you don't have stability. So don't. Normally, it's good in the studies to have a pre-session where you check this, because normally if the person comes like doesn't change, the conditions that don't change a lot, so the motor threshold will not change a lot. Yeah, if I do my pre-test today and tomorrow I do my stimulation protocol, because for example, this is something I do. We are testing certain protocol, and I don't want the brain to be excited before or change before. I want to test what is happening with this protocol particularly. I don't want to send extra pulses. I don't want my brain to change. So the day before, I find the motor threshold. Next day, I will apply my stimulation with this threshold I found. Not 1 month later, around the same time. Yeah? Okay. Any questions? Are we okay? Anyone want to go to the bathroom, take a small break or something? We are going to talk then now about the equipment. Okay, so nobody. TMS equipment. So, very important, machine coils and the TMS pulse. And it's important because depending on the equipment I have, I will also get some outcomes. Yeah? This is a model of the TMS machine. Okay, 1 second. So we'll see that there are different shape pulses, right? And for different shape pulses, you will need different stimulators, or the mechanism inside the machine should be different. Here, you have a biphasic stimulator. So the pulse of this machine is biphasic. It could be monophasic, biphasic. Monophasic, biphasic, polyphasic, and there are machines that have like very fancy, other fancy shape of the pulses, but for the biphasic stimulator, is the most common, or the monophasic. You will have a different system to generate this pulse that you want. So basically, like really summarize, you have here, you need just to have this thing here. And the current passes, and then the condenser, he will have a capacitor that is charged, and then when the switch is closed, the current will pass, and this is a biphasic stimulator, so you will have the two, actually I should have done this before. What is the shape? No, like like a sinusoid wave will come through the inductor. So the magnetic pulse is generating the coil. Once this is charged, the switch is closed, the pulse passes one part and then the second part, right? It goes through the coil, one part and then the second part. But when I have my monophasic, there is a second circuit here, you see here, where part of the second part of the pulse, of the signal pulse, will not go, will be lost in this area here. So I will always have the first part of my signal pulse. This is cool to know because the pulse that I'm applying will have different effects on my TMS outcome. You need to know that and you need to report that. When you are going to do your study, it's important that you say which type of pulse you use so people can reproduce it. We will talk a little bit about this. So I just wanted to show, and I wanted just to make you aware of this, that you need to know what type of pulse you are using, and that each machine has, depending on the pulse you are using, will have a different circuit. And when you have both, you can have both, so you will have a click when you are changing the type of pulse shape, so you cannot use the same machine for biphasic, for monophasic, you cannot. So I just want to make you aware of it. You have different type of machines. This is a McVenture machine for, like, you can generate RTMS or single pulse. This is a machine I'm working now with. This is a MaxStim machine. I think this is the B-Stim to generate 2 type of pulses, and I'm not sure if this is the RTMS, the RTMS machine, the rapid square. So you can generate different protocols with this machine. I worked with it long time ago. Okay, you have different type of machines. When you are writing your study, you need to report which machine you used, because for every machine, it will be different, yeah? Okay, and also, well, to make you aware of this, this is for certain type of stimulation. This is with this machine. I like it a lot because with this machine you can do almost everything. You can have this. With MaxStim here, you need this machine to do per pulse protocols. We will talk about it. And then you have this machine to do repetitive. And with this machine here, you can do everything you want. Single pulse, per pulse, you can create anything you want, which is kind of cool, right? You have one machine and then you can do everything. The TMS coils, very important. There are different type of coils, and each of them has different possibilities. So I thought it was very good to mention it, because when you are going to read the papers about what they did, so it's very important that you see which type you always, when you see the methodology, you will see type of machine, type of pulse, type of coil, and they also say the size of the coil. Yeah. So all these details are important because TMS response will depend on all these parameters. So it's not enough. It's very interesting when I have seen people that they just get a machine and they want to do TMS and they just get the machine and they put it and that's it. I think you need to know all this kind of stuff to know what you are doing, and then you plan also accordingly. So I'm going to talk just about the most common coils. One of them is the circular coil, and the diameter is between 8 and 15 centimeters. You need to report that when you are doing your study. Also, what type of brand, which brand you use, because every brand is different, so you need to say that. The maximum induced current is at the other edge of the coil. It means you have a response is more like around in the end. Activate superficial cortical layers. It means neurons located at the depth of 10.5 millimeters from the surface of the skull, and is recommended for large and superficial motor areas such as upper limb motor areas. So depends on what you want to do, you might want to find the type of coil you want. Here this is a cognitive lab, right? Like you do mainly cognition. But in motor cortex labs, or when you work mainly with neurology, you will have all type of coils. All the coils we have seen here and more. You will have all type of coils because you need different type of coils for different type of stuff. And they will always be trying to get improvement with different type of coils. You have different depth, you have different response, you have different areas of stimulation, so it's very important. Figure of 8 coils are like the most common. Like normally you see like everybody use it. Why? Because it's more focal. The response, we will see a small figure about it when they have done modeling, trying to show what the difference between using a circular coil and the figure of 8, which are the most commonly used. So the maximum current happens at the intersection. So here, where both coils meet. So the electric field is at maximum under that center of the hotspot. So I will locate my coil on the hotspot. The center of the coil will be located on my hotspot, or the stimulation point I want, and is recommended for more accurate and defined areas. Stimulate neurons located at the depth of 11.5 from the surface of the skull. I wanted to say something else here. Let's see if I found it. No. Let's continue. Double cone coils. The stimulation of this coil is not that focal, but it reaches deep cortical layers. So you sacrifice focality for depth. So you need to know what you want. So you can stimulate motor areas of lower limbs, which are very hard to locate. You can do it with the figure of 8 coils. I have seen some people fighting with it. It's horrible to try to get it, but you can get it with an figure of 8 coil. But then if you use a double coin, probably would be easier. So stimulation reaches broad areas of M1 and activates bilateral lower and upper limbs, and also facial muscles. So you get facial contraction. But of course, with the figure of 8 coil, you can also get a pretty hard facial contraction if you put it in the frontal area. It's kind of painful. I think maybe I'm too coward and I cry easily, but you can get like really facial contraction there. But okay, I guess it's when you are stimulating. I haven't used this type of coil. I have seen it. I haven't used it. When you use it probably to stimulate other areas, it already reaches facial without the need of being in the frontal region. H coils. I have seen these coils. I have never used it. It has obviously reduced focality and are bigger than the conventional coil, like really bigger, like a helmet. You see, this is a helmet. And has lower electric field alternation with depth, so is recommended for deeper and broader prefrontal brain regions for depression studies or things like this. For therapy, you might want to use it. I haven't used it. I have seen them. It would be interesting to test it, actually. I have never tested it. When I just started with this, I wanted to test all type of stimulation all the time, but then I'll tell you the story, short. They wanted to, we were in this course, like I was just 1 month. I always work with stimulation, but I work with stimulation in cells and in like all type of things. And then when I jumped to humans, which I wanted, there was just exactly at this congress, and they were showing like we are doing here, like some talks, and then the hands-on, and they're like, yeah, who wants to be a volunteer? And I wanted to be the volunteer, and then they started to do different type of stimulation with me. And then they tested the double stimulation, and it was so painful in the frontal region. It was really, really painful. I stopped being a volunteer. It really was like, oh, it's painful. No. And then he continued stimulating, and it was really, really painful. So when I saw this, and I knew it was very deep, so I thought, no, I pass. Okay, we have other type of coils which are very important. You have also here, you have this here, an example of the cool coil. This is very important when you are going to use RTMS protocols, because the coil gets heated. So you don't want that, because then you will have to modify. If you don't have a cool coil, you might want to modify your protocol, because the coil will get really warm, and if the machine has safety parameters, it will block the machine, and you will not be able to continue with your simulation. I didn't have the experience that I was working long time ago with that, but I didn't know this. I was just learning and playing a little bit around. So with an RTMS protocol, I think it was 1 hertz. And it means each second a pulse is being sent. And I was using the testing was, the threshold I was using was quite high for the person I was playing with. So it was a colleague. We're testing. You need to test the protocol. And I'm testing it with myself as well. When you are going to do a study, you need to know it's good that you know how the person will feel. So that's why I know this stimulation in the frontal region, because I have applied to myself when I'm testing, like, it's painful. So I know, and then you can tell the person, yeah, you will feel this. Anyways, yeah, the stimulation is around 15 minutes, the protocol. We will see. I have stored here, fixed here some protocols that you can use. And then we will also learn to write protocols here in this machine. So for 1 hertz, for example, you will need to, at 7 minutes, the machine stopped because it was too warm for the safety parameters of the machine. 7.5. So I want to do like a kind of pilot, and then I just did the pilot the half because I couldn't continue. Or in other occasions, I was trying to use like ice. I don't know if that's correct. So trying to see if I can just make it cool. But you don't need, it was just during playing to try to get to know the equipment, how it works. So, but then fortunately in the labs where I have been, they always have equipment without cooling system and equipment with cooling system. So when you are gonna do RTMS, you need to work with the cooling system, okay? So this was in my early times when I just wanted to know how everything worked and how the machine was doing and trying to learn how to program. So I learned that actually you do need to, use a cooling system. Okay. Oh, sorry about that. I checked reference. I just want to put here there. Okay. This should not be here. I thought I changed it. Okay, we'll leave it here. So it means that your sheets, your also is a check reference? Oh, I'm sorry about that. I just wanted to, yeah, I put a lot of references down. It means this is not the right one, probably. And I just was looking for different figures, and I was like, okay, this is the figure that shows better what I want to show, and then I changed it like last minute, so I'm sorry about that. But this is a good moment to tell you that I checked a lot of literature, and I tried to put all the literature in that I'm using, all the figures, everything has like the literature that you can check, so that you have a starting point to check for information and all this kind of stuff, so that you don't have a big list in the end, but every figure has the reference. And then I thought it was quite kind of cool that you go and then you check, okay, this person, and then you find this figure, and you find what I'm talking about. Yeah, so I'm sorry about that. But what I wanted to show you here anyways, ignoring this, is like, you see the figure of the circular coil. This is how the induced electric field will be. So it will be strong in the border of the coil. Yeah? So the effect is completely different to the figure of a coil where the strongest part is in the middle of the coil. And you have also some effect in the borders, but the strongest part is in the middle. So when I'm stimulating with a figure circular coil and I put the coil here, the part will be stimulated is mainly around. When I'm using a figure of a coil, the part that will be stimulated will be the middle of the coil. You see the difference? It's really more focal. So that's why it's kind of the preferred stimulation coil. So double coils induce their highest eddy currents where the winding joins improving stimulation accuracy. But circular coils induce the highest eddy currents under the winding and no current under the center. So you don't get any current in the center for the circular coils. And here I just put this, like the different type of coils you have, and you have these are models of how the brain responds to the stimulation. So normally we will be moving in this area here for the figure of a coil, you see here, and or the 8 coils, and then you see that is really focal where I'm stimulating, you see. So I just put, I know it's like a very big thing with a lot of circles and all this kind of stuff, but I thought it was kind of interesting. So you could see the focality, which is very important. It's very important, and we will see if you want to work with do studies about, you want to investigate a region, right? Frontal area or visual or whatever. So you want it to be very focal. You want to stimulate what you want to stimulate, not all the brain. So if I want to stimulate with the circular coil, look, I'm stimulating like a big area. But if I want to if I want to check how the lateral prefrontal cortex changes, or if I want to create a virtual lesion, we will talk about it. So you really want, and you say, okay, I want to check connectivity, right? And I want to see how the brain reacts if I switch out this area. So you want to really be focal and just target the area. Yeah? So that's the idea of this example here. You see how
Excuse me, for our type of cornea, so which induced the electric field would that be in the brain? So just for this one.
this will be in this range.
Okay. So
it's
it's
very
quite
common.
focal.
Yeah.
Okay.
You have a quite cool equipment here. It's very easy to, well, it's not very easy to use, but
Well, you need to know how to use it.
no, it is easy to use. It's just like, that's why I haven't worked with this equipment before, and then it took me a while to get it. But it's really, you have a lot of possibilities with it. It's kind of cool. You should really try to take advantage of it because it's very modern. You have a lot of possibilities. It's so make business with Alex or the system or the people that are working with this, that they know about it. It's kind of cool. And you have a lot of possibilities. You can apply like focal stimulation. You can do a lot of stuff. We will talk about it tomorrow. Okay. Let me see. Okay, so this single pulse TMS is to make an introduction to the shape pulse, which is so important. So single pulse TMS is like the simplest thing you can do for TMS, and tomorrow we'll see that you can do a lot of stuff with just one pulse. Well, no one pulse. You have a collection of pulses, right? But you can do study. And so it's available in all TMS devices. It's safe to use. With single pulse, you will know. I mean, I guess you have a little bit of. Have you heard a little bit about TMS, that it can create seizures or a thing like that? We'll talk about it in a moment. This goes until 1. Until 1 is the class. Oh my goodness, no. So we might need to.
Ask you a question, because it is like the TMS has mentioned that there are some of the persons who that they are not fit to use a TMS, but then if they have not declared, if they just hide it, and by the time that it has gone, so how can we be faster?
I didn't understand. Sorry, can you?
I mean, it's like TMS that we have a series of questions that you have to go in and check whether or not you have got this kind of like previous brain coma before or something like that. But if they just they hide that or they ignore that and then they just tell that everything is all right, so.
Yeah, well, yeah, actually, I think we might need to use part of the afternoon to talk about it. It's really important. I thought I would be quicker with this, but that's very important. What you need to do is to scare the subject. You need to, you have your consent form to scare the subject. You have your consent form. In your consent form, it says all the effects that it can have. And I always when I am doing experiments, I always try to confirm that it is so, but now in Duke, it's quite strict. So you have someone who is doing it's very long, the process to get someone is really strict and really very, very, how you call, organized. It's kind of cool, very strict. I have always been strict, but they're really very, very, very well established. So you need to be very clear with the subject what the effects could be. When they are filling the form, you need to be clear. Do you have headaches? No, because I can give you headaches. You have migraines? No, because I can make your migraine worse. You have epilepsy because I can cause you a seizure. It's very important that you say, I can do it.
The one who conduct the TMS should know, and for example, the first aid for the seizure, for example.
Yeah, this is important. We will talk about it. I think, it's good to know this stuff I'm telling you now because it's important for your experiments, but the safety, that will be the next, if we need to get some hours, for 1 hour for them, we need to use it because it's really, really important. Some things you need to organize here in the lab, in the groups. Some other course, so after this course, you need to have an extra course about seizure management. I can give you some ideas about how to, the most common things. We'll talk, but I don't want to give you previews. I will tell you so you come back in the afternoon, because that's the nicest thing when you are really in action, right? But the most common thing is no seizures for me. If you read the literature and everything I put there, they say headaches. Not my experience. My experience is syncope. People start to black out. This is the most common for me in my experience. I have had this a lot. You need to learn how to manage that. And then for that, I can tell you more or less how we can do it, but I'm not a neurologist, I'm not an expert. So what you need to do is to bring someone, an epileptologist, better, an epileptologist who will give you a talk and will teach you what to do in these cases. For a seizure, the first thing I will tell you, no panic, and they will tell the same. And then we will, in the practical part, or when we talk about the safety, we'll talk about this. You need to be calm, like nothing is happening, like you know what to do, although you don't know, you will know how to do. You need, when you are going to do TMS, you need to know what to do. That's very important. But you need to keep calm. No, there is a, no, you calm, okay, yes, so you see it, you recognize it, and you proceed calmly. The only thing you can do is just calmly react. The person has to be in certain, or put it down, or just talk calmly, or I'll explain you that, yeah? Important, you need to make emphasis on what you can do. If they lie, that's very important, yeah? I think people normally are afraid of it, so they will not lie. You need to, I know that for some people could be tempting to hide, you know, like in the university where I am, I think they are really emphasize a lot the effects. And then I'm thinking, oh, people will not come because it's so emphasized, you know, like it's so clear, like, oh my goodness, no, if I read this, I will not come because I can have headache, I can have a seizure, I can have this, I can have tinnitus, so I will not come. But it's important because the people that come, they will really want to do the study, you know. So you should not hide it. You should tell all the possibilities. We'll talk about it. Okay, let's run. Let me see how far we are. Oh my goodness. Oh no, yeah, we are almost finishing. Okay, okay, this is very important because depending on the waveform you are using and the direction, you will have different effects. If you see different papers and they are using monophasic or biphasic, the results could be different, probably be different. And you have also polyphasic. So monophasic is used for more accurate than biphasic, has lower noise and lower heat because it's only the half of the wave. Yeah, so this is the monophasic. Remember I told you that the current is sent and then part is passing and the other part of the circuit is just eliminates the other half? When I was showing you this here, so the current is passing the same shape, but then just half is lost in this half circuit. What doesn't have, what it has against, it's not easy to obtain bilateral cortical responses. So you need to check when you are doing your study what you want and really check what the literature says about these things. But okay, you don't have a lot of options as you have here a biphasic simulator, which is normal and it works for everything. So it's kind of cool. For biphasic is used for short efficient pulse. Suited to bilateral cortical stimulation, but has higher noise and possibly is less accurate than monophasic. The advantage of monophasic is that you can kind of determine it's more precise, so you can determine the effect of the pulse. But here you have a double, like an upper part and a lower part, so the effects are more shady. Like, okay, you don't know which part affected what, what did what, but with the monophasic, you have just one phase, and then you know that this part did this effect on the cells. Clear? But okay, this is very used. Also, for monophasic, you need a little bit more stimulation. Sometimes it could be stronger. So in some studies, although I have read that they use monophasic just with the kids, I was using biphasic because it was just the lower intensity. So you want to use the lowest possible. And polyphasic, it's just efficient. It's efficient, suited to bilateral cortical stimulation, and has a very high noise, and also is overheat, and is less accurate than monophasic. So we have here different ways to see some studies that have been done, trying to check what's the difference between the different pulses. And we have here half sine, so it's a monophasic, half sine, and full sine. And this is on checking the amplitude, the MEP's amplitude as a function of the stimulation, the resting motor threshold. And as you can see, there are differences when I'm using monophasic or different type of shapes. So it's very important that I take this into consideration when planning my study. Okay? I think I did not put a lot of, give me 1 second. Let's see. Okay, this is very important. This is what I want to show you. If you want to say, so message, take home message for this is, the effect is different, so that's very important if I use one type of the other. And the other message is here. What is the resting motor threshold based on maximal stimulator output, checking the different shape currents. And as you can see, the biphasic has lowest intensity. That is what I mentioned before, right? Then it could be nicer to use that one because higher intensity is also more painful. So you want to probably take that into consideration. So this is one of the things I thought it was very important to show. Yeah. And yeah, in general, I think this is the most important thing that I wanted to show you. But you have, yeah. Here, I'm also showing the current direction. And I should have drawn something like this. The current direction is, let's see, I can show you here with the coil. You have the coil here. If the current goes from here to there, or the opposite, yeah. Posterior, anterior, posterior, anterior, or anterior, posterior. And if you see this figure here, you can see, here, you see that there are differences when I'm using even for the same shape of the current. There are differences if I'm sending anterior, posterior, or posterior, anterior. The response in the brain is different. So you need to take also that into consideration, and you need to report what was the direction of the current. Okay? So if you have here is the latency of the MEP, so it means how long it takes for the MEP to appear. For a monophasic, for anterior posterior, it takes longer time than for a posterior anterior. So from posterior to anterior area. For the biphasic, the change is not that big. But this is very important to know. Also, very important, when you are changing the direction, not only the direction of the current, but the direction of the coil. Normally, when we are doing motor cortex, we have like 45 degrees from this quadrant. But if you change it like completely perpendicular to the midline, the response will be completely different. So this should be also taken into account. Yeah? A stimulation side, and so the stimulation side, we have been talking about this a lot. So we have motor cortex and we have other areas outside motor cortex. Motor areas could be hand, knee, mouth, legs. It's kind of cool. I had a colleague that was doing a study moving with the stimulating the mouth. It was kind of nice. Difficult, I guess, but nice. And no motor area like the dorsolateral prefrontal cortex, visual regions, or other somatosensory areas. I want to show here like the most common for the hand. Well, for motor cortex, we study a lot the hand. So this will be the FDI. I also made an error here. My goodness. Sorry about that. Just I wanted to make sure that I will give you the right references. It's mainly for the references because I changed several times the thing and I just wanted to give you the right references. So here I'm showing you the areas we want to stimulate. So where do you put the electrodes? Right, so for motor cortex, I'll put my electrodes here in the hand, but I will stimulate the head, okay? So the regions we use the FDI, the ADM, and the APB. So this, or this. Normally I have always worked with this one. As I have worked with the FDI, so I always do that one. But you have other alternatives. And as I said, you can also stimulate legs, you can stimulate other regions. Non-motor areas, you can stimulate non-motor areas, and there are different ways that you can find the location of these non-motor areas. Here you have a neuronavigator, which is kind of cool. You have the brain site. You have many possibilities about how to locate the region of stimulation. Suppose you want to stimulate the dorsolateral prefrontal cortex or Brodmann area 9. Are you familiar with Brodmann areas? Brodmann, like the brain, you can divide it in different areas, and Brodmann is one of the templates you can divide it. Yeah, and every region has a different function. So you suppose BA9 will be one of the like memory areas, or BA46, for example. So you say, okay, I want to study memory, right? You say, I'm going to check how memory changes in BA9, Brodmann area 9, for example. I'm going to stimulate there. How do I find BA9? Because I cannot see, so you have different possibilities. You can do it through your navigator. You can send the person to the scanner. They will do a task that is related to memory, the task you are of interest. The person performs the task, you analyze your data, and you see an activation there. And this activation in this Brodmann area, I get it here. I feed the neuronavigator with this, and then I put my code where the navigator is telling me. Do you understand that? Or another way to find, for example, my stimulation point is for the dorsolateral prefrontal cortex is the method, the 5 centimeters method is more or less. So, and it is, you find the motor cortex, you find the motor area, and from there you measure 5 centimeters frontal. And then that will be your dorsolateral prefrontal region. Another way to do it, so I'm giving you 3 ways to do it here. Well, 3. Is using the 10-20 system. Are you familiar with the 10-20 system? With the electroencephalography, in electroencephalography you have to put electrodes, right? And these electrodes are located not in any way. They have some system. One of the systems you use for EEG is the 10-20 system. So it's measuring between 10 and 20 centimeters or something like that. So you can also try to, these electrodes are related to some regions in the brain. What you can try to do is locate the DLPFC, which will be located between F3 and AF3. Then you calculate this area measuring with a measurement tape. You start to do all the calculations. It's very easy to do if you follow the instructions how to do it. And then you can locate for everybody. It will be individualized, of course. And for everybody, when you start your session, so you say, okay, I don't have MRI, I don't have navigator, I don't think this method of 5 centimeters is that good. So what I would try to do is use that 10-20, my 10-20 system. So you get your measurement tape and start to measure and locate the position, the site, simulation site. This is TMS stimulation with the Neuro Navigator. So I was doing a study with MS patients, and we wanted to stimulate Brodmann area 9 and 46. That's why I just mentioned it. And the person went the first day, the second session they went to the scanner. They did a task, and then when they came out, I analyzed the data, and I saw for every person where was BA9. So you have two possibilities. You can either get the functional image and feed it to your, this is not the same navigator. This was another navigator, not this. You can either put this, the functional image with the structural image of your subject, individualized, or you can get the coordinate of this activation and feed it into the navigator using the structural image of your participant, and you use the coordinate. You don't need to use the whole, you don't need to use the functional image. You can use also only the coordinate. Another option is I don't have the money to take the person 3 times to the scanner, so but to have it 1 hour, but I can have a structural image that is just 5 minutes or 10 minutes. So I get that image. And in the literature, it says, well, you can stimulate 46, 30, minus 3. So you feed this coordinate and put it in that is located on the brain. You have many possibilities. And then everybody will be stimulated on the same coordinate, but you have the individual structural image. Clear? So this is what I was telling you. You can either do functional, or you can just select your coordinate, or you can go to the literature and say everybody is saying, or most of the studies are showing me that this is I want middle frontal gyrus. So the coordinate for the right will be 48, 31, 42. So I feed the navigator with this coordinate, and it will be located there, and I put my code there. Okay, so we finished this part. TMS induced response depends on several factors. Just to summarize: stimulation site, coil type, see if it's a figure of 8, 8, or whatever, coil orientation and tilt. It's very complicated, this thing. So it's not only like I have the right orientation, but also the tilt should be correct. Magnetic field a waveform, monophasic, biphasic, multi. I put multi here like because the biphasic like several, the polyphasic is like several biphasic, right? Pulse direction, if it is anterior, posterior, posterior, anterior. Current intensity and dose. What is the intensity of the current and how much I'm going to apply? Frequency and pattern of stimulation when I'm using RTMS. So you use certain frequency and different patterns. We will see that tomorrow. Tissue properties, conductivity and permittivity. You cannot measure that, but the E-field modeling that we were talking before, they do calculate. They try to calculate what is that, right? But this affects. We don't use it, but it affects. So some people are respondents and some people are not respondent. Probably that's one of the reasons. The tissue properties of the person. Interaction coil tissue, which is unique for each subject. So that's why probably you have groups of respondents and some people that don't respond. And brain state. If the person is sleepy, if the person is tired, if the person is very excited, so you will have a different response. And that's it, the first part. Oh my goodness, that was a lot of information. I hope it was good for you.